PTSD occurs in about 5 to 10% of the general population and up to 25% of highly traumatized civilians and veterans. It is also one of the leading causes of suicide and comorbid depression and substance abuse. Despite this, we only have 2 FDA-approved drugs, both SSRIs approved over 20 years ago. What can we do about this? As you know, psychiatry is probably the last frontier for next-generation biologically driven therapeutics, and we feel that post-traumatic stress is particularly tractable in this next generation for several reasons. First of all, the genetics of psychiatric disorders broadly, and PTSD specifically, has been transformational in the last few years, now with GWAS studies of over 1 million people leading to almost 200 gene candidates. From a neuroscience perspective, PTSD is focused on a neural circuitry of fear, and particularly the amygdala, that is conserved across all mammals and most vertebrates. So we have an enormous amount of decades of research in understanding the biology of trauma. We also know for PTSD, unlike most other psychiatric disorders, we know when it starts. By definition, it starts at the time of the trauma, and that gives us enormous power for both understanding the biology longitudinally, as well as predictive biomarkers, and for the possibility of prevention, which I will focus on today. Finally, while you've heard a lot about learning and memory disorders such as dementia, PTSD seems to be the opposite, where you overlearn the initial trauma and have difficulty regulating it over time. If we could interfere with that initial learning process, we could block consolidation and prevent the development of PTSD. Our goal is to develop a molecular neurobiology of post-traumatic stress, leading to predictive and treatment biomarkers, leading to novel interventions for prevention and treatment. The gap is as following. Right now, if you have chest pain, you know to go to the emergency department, and within a couple hours, based on physiological biomarkers such as an EKG and blood-based biomarkers such as cardiac enzymes, you can have interventions such as biologics to break the clot, or interventional radiology that can prevent the long-term sequelae of myocardial infarction. In psychiatric disorders, we do not have this. While you can have a severe physical trauma, you can go to the emergency department and you can get psychological support, but we do not have the biomarkers for predicting what 10% of the people are going to later develop PTSD, nor do we have those biomarkers or tools for neurobiologically based prevention or intervention. This is despite the fact that memories do not form immediately, rather they form over a period of time. On the bottom, we show the pathways of memory consolidation. We know that memories start in an early plastic period in which they're plastic for minutes to hours to days before they become permanently embedded within the brain. Our focus is on this memory consolidation process. In the upper right, you see the main circuitry involved in fear and threat processing. Particularly the amygdala is conserved across mammals and is very similar in its cellular and molecular composition in mice and humans. And we know that when that initial trauma occurs, synaptic events occur in the amygdala that lead to changes that then lead to long-term fear reflex processing. And more so than any other place in psychiatry, we know the neural circuitry of this panic-type fear reflex. Downstream projections from the amygdala lead to the increased heart rate, blood pressure changes, respiratory changes, GI distress, and the adrenaline and cortisol response of acute stress and trauma. To understand better the biomarkers in humans and the longitudinal development of these biomarkers, we have embarked upon a number of studies over the last decade, starting with work in Emory and Grady Hospital in Atlanta and Miami with about 500 participants, and then that expanded to a multi-site study of over 5,000 participants in emergency departments around the country. We enrolled patients immediately after car accidents, car crashes. We took blood, we did physiological measurements, quantitative psychological assessments, embedded digital biomarkers such as wearable devices and phone-based markers, and then brought people back at several different time points for neuroimaging and other brain-based biomarkers and followed them up for 12 months. This allowed us to develop predictive analytics so that we can say with about an 85 to 95% likelihood who is going to develop PTSD in the early aftermath of trauma, as well as predictive response biomarkers. One of those biomarkers, as I've mentioned, is the amygdala, which is conserved across species and where I'll talk more about our discovery. But specifically with the amygdala, we showed that even at 2 weeks after trauma, notably we don't diagnose PTSD until 1 to 3 months following trauma, but even in the first couple weeks, amygdala activation in an MRI scanner to fearful cues is highly predictive of later PTSD at 3 months and 12 months later. Other studies have shown that the amygdala hyperreactivity normalizes after treatment, so it seems to be both predictive and treatment responsive biomarker. In terms of scalability and rapid biomarkers, we've shown that measures of the sympathetic nervous system and adrenaline, for example, the sympathetic skin conductance response, is highly predictive. In the first hour or two after trauma, if one is measuring galvanic skin response and asks a patient, what brought you into the hospital, that adrenaline response is highly predictive of both symptoms and diagnosis 6 months later. So we now have treatment and predictive biomarkers, as well as a whole host of quantitative assessments and other blood-based biomarkers that are being developed. In parallel with this, for the last decade or more, we've been focused on understanding the biological basis of fear consolidation in mammals, specifically doing differential gene expression and other approaches in the mouse amygdala, following a model of trauma, specifically Pavlovian fear conditioning. In screening these genes, we find a number of very interesting pathways related to synaptic plasticity, but particularly here we show tachykinin 2, which encodes the neurokinin B gene. And tachykinin 2 is expressed at a temporal way that's very consistent with consolidation, up at 30 minutes and back down to normal at 2 hours. Importantly, it's expressed in the right specific cells within the central medial amygdala in green here. These are the cells that project to these hardwired downstream subcortical regions that mediate the fear and threat response. So it has the right temporal action, it's expressed in the right place, and then if we give an antagonist, a tool compound called asanetant, directly into the amygdala or peripherally in the immediate aftermath of fear conditioning up to several hours later, and this shows on the right animals' fear response or freezing response after 5 tone shock pairings in which they've been given this antagonist up to 1 to 4 hours after the trauma. We then measure their level of fear or trauma responses days later, and those who receive the NKB antagonist show significantly reduced fear. So that suggests necessity. If we look at sufficiency in terms of overexpressing tachykinin 2 with genetically modified viruses, we get significant increases in fear. So we can push it down, pull it up. If we overexpress the gene and then block the receptor with the antagonist, we normalize fear. Other studies have shown in our work that if we can, using modern day neuroscience tools, can overactivate or inhibit these same TAC2 cells and increase or decrease the fear response accordingly. In parallel with the emergency department studies and other collaborators, we have DoD funding now to use these emergency department networks where we can combine predictive analytics with interventions of novel approaches to preventing PTSD. And again, the idea is we could eventually have the same protocols that we do for heart attack or stroke, but in the emergency department or on the battlefield to prevent the long-term sequelae of PTSD. We've also recently collaborated with Atomwise to use AI or machine learning for the predicted structure of the NKB receptor or TAC2 receptor and identify specific antagonists from a library of over 30,000 compounds. From those, they then identified the top 100 predicted compounds, and we tested them in vitro in a Presto Tango GPCR-specific assay, identifying a number of new compounds that are much more potent antagonists than the tool compound asanetant. We are now working to expand this. So in summary, we've developed a rational neurobiology approach to post-traumatic stress prevention. That could be the first time that we could have a targeted prevention of a psychiatric disorder informed by deep understanding of the fear consolidation process developed over several decades. There are multiple ways to interfere in fear consolidation pathways following trauma. We've demonstrated efficacy of NKB identified from observational differential gene expression and then multiple mechanistic studies. A lead compound has been identified, and we have a method of use patent for general NKB antagonists, as well as are working towards composition of matter IP for some of the new compounds. We've identified also several additional alternative mechanisms and potential future pipelines for targeting the molecular mechanisms of trauma consolidation. We're looking for partners and collaborators for next steps, including lead optimization of our primary hit, as well as others, as well as biomarker development for biomarkers for treatment and predictive outcomes. All of these, we believe, will significantly de-risk clinical trials in psychiatry. Thank you so much for your time.